Middle of the week, hump day. How you guys doing? Better. Better than what? Better than yesterday? Biochemistry makes things better? No? I hear some dissent in that. I don't know. I hope it's making things better for you. I'm going to continue today and hopefully finish up with our considerations of the sort of elements of protein structure and some things specific to that. And then on Friday, I plan to start talking more specifically about some of the applications of that knowledge. So that will be something that will hopefully resonate a little bit with you after you've learned the things that we've talked about here. Last time I talked about mostly primary, secondary, and tertiary structure. And today I'm going to finish that off, talk a little bit about quaternary structure. And you'll see quaternary structure is probably one of the easiest ones to understand. And then we'll talk about some of the other ramifications of that structure and talk about a very, very important process for proteins, and that's the phenomenon known as folding. And folding is a really amazing process, as I hope to convince you today. So the first thing I want to talk about is something I've alluded to on a couple of occasions, but I haven't explicitly shown it to you. And I think it's a pretty critical component of protein structure. And they're known as disulfide bonds. So disulfide bonds are, you may recall, covalent bonds that form between the side chains of cysteine residues. Cysteines, you should remember, have a sulfhydryl, that is an SH, as a part of their R group in the amino acid. And two SHs, if you put them very close together, will basically oxidize and form a disulfide bond, meaning an SS bond splitting out the hydrogens in the process. And so that covalent bond turns out to be something that is a very, very strong structural component of some proteins. Now, you're going to see this protein I've got on the screen today on 2 or 3 occasions, so I'll get you warmed up to it a little bit. It's called ribonuclease. And ribonuclease is an enzyme that is ubiquitous among living organisms. It is an enzyme whose catalytic activity is aimed at degrading RNA. Okay? You might think, well, why do we want to degrade RNA? We'll talk about that a little bit next term, but most importantly, we degrade RNA because RNA can code for proteins, and we don't always want to make all the proteins all at once. So being able to degrade it is one of the ways we can stop a protein from being produced. And RNases are very important players in that process. RNases, and by the way, ribonuclease is called RNase, R-N-A-S-E. RNases are, in many cases, really interesting and important proteins. These enzymes can, in fact, catalyze, in addition to catalyzing the breakdown of RNA, they're very stable. Now, what I haven't told you too much about so far is that the protein structures that we've talked about, hydrogen bonds, for example, are the elements that are the forces that are holding together many of the protein structures that we have seen. And those hydrogen bonds are relatively weak. And they can easily be overcome with heat. All right? So today I'm going to talk a little bit about other forces that help stabilize proteins. And one of those is disulfide bonds. So disulfide is the strongest of the bonds that are stabilizing tertiary structure. Okay? So tertiary structure has a variety of forces that help give it stability. Hydrogen bonds are one form, and disulfide bonds are another. And the disulfide bonds, as I said, are very strong. And I like to think of them, if you go into a building and you see support beams. You can see support beams in here. There's a support beam. There's a support beam. There's a support beam. They're critical for the overall structure because of their strength in holding up this room, hopefully. And these support beams here in this protein are also very important for their strength. Okay? Now, in contrast to these support beams, which are pretty critical in holding up the room, all right, and might survive an earthquake fairly well, these guys, these disulfide bonds in some proteins really help to hold a protein together under very difficult conditions, and other times, most proteins, they don't. So I'm not going to go into that in too much detail, but suffice it to say that they are nonetheless important components. All right? Well, first I'm going to talk about these disulfide bonds, then I'm going to tell you a little bit more about some other forces that stabilize the tertiary structure of proteins. All right? So we see this ribonuclease that has these disulfide bonds, and these disulfide bonds arise because the structure of the protein has folded to a point where the SH of one cysteine is brought into close proximity of an SH of another cysteine, and from what I've told you so far, getting them into close proximity is sufficient for them to react with each other. All right? So they're brought into close proximity, they react, and they form that structure. Okay. When you see the term native, it means active, normal, whatever you want to call it. It's the way that the enzyme is made and folded, and this enzyme, if I took it and I mixed it with some RNA, it would, in fact, degrade the RNA very readily. Okay? Very, very readily. If I take ribonuclease and I boil it, ribonuclease has an interesting property. In contrast to most enzymes that lose their activity when you boil them, ribonuclease does not lose its activity. When it cools back down, it's just as active as it was before you started. One of the reasons that happens is that the disulfide bonds in ribonuclease help to stabilize it even under high temperatures. Most other enzymes, even though they almost all have disulfide bonds, most other enzymes don't have that property. Ribonuclease does have that property. So we think it's because these bonds are in very critical places that really help to hold everything else into place. Yes? Good question. Does it work at the high temperature, or does it work only at the lower temperature? Ribonucleases can work in relatively elevated temperatures. They can work up to, say, 60 degrees centigrade, where most enzymes wouldn't make it to that. They're not active at 100 degrees, but when you cool them back down to 60, they're active again. So that sort of tells us that there's other considerations of structure that help give rise to function of proteins. All right? We'll talk a little bit about those in just a bit. Now, what this figure is showing you is, well, is it possible to break disulfide bonds? And if it is, what happens if I break the bonds? Well, if there are chemical reagents that I can use to break disulfide bonds. One of the chemical reagents is what you see on the screen there. It's called beta-mercaptoethanol. And what it does is it converts an SS bond into two SH bonds. So they come apart. Okay? Well, if they come apart, it would be kind of like taking the beams in this building out and then putting an earthquake and seeing what happens. Right? If we did that, this building or this room wouldn't be here for very long. And similarly, if I take these beams out and I boil this guy, or I use something that destroys hydrogen bonds, urea is one of those things, either of those will cause this protein to completely come apart and not be active. When you do that, you have done what we call denaturing of a protein. We have denatured it. Denatured it, you can think of as unfolding. And when you talk about a denatured protein is inactive. This protein on the right, the denatured, reduced ribonuclease, will not, in fact, catalyze the breakdown of RNA. Now, I'm going to say more about this protein in just a little bit, but I wanted to give you some background about this because it's going to come up later in the lecture again today. Yes, question? Yeah. Okay, she got a very good question. Okay, this is what I love. Okay, you put this up here and people get thinking about it. Seriously. All right, she's thinking. Well, if they started out with an SH, and if I convert them to SH, what keeps them from going back into their original SH form? Okay, so she's actually getting ahead of me in the lecture. So there's two things, but I will answer your question since you've asked. Okay, one of the things that happens with this is the fact that we're boiling it and we're driving them apart. The other is if we want to keep them apart, we need to keep a sufficient supply of the beta-mercaptoethanol in there so we keep them in the reduced form. If we don't have sufficient amounts of this in there, we're going to see that this guy can come back together. And it's an interesting problem of it coming back together. Good question. Everybody got that? Yes, question back there. Oh, boy, very good question. What about conditions of very low pH? What do you think? Probably same effect. For most enzymes, and again, this is not an absolute rule, but for most enzymes, changing the pH drastically will also unfold the protein, because in that case, we're disrupting ionic interactions. When we change the pH, we're adding a proton or we're taking a proton off, depending if we're making a low pH or a high pH, right? Imagine, if you will, that I've got a protein structure that is folded up, and I'm thinking of the forces that hold it together. So here's, let's say, one strand over here in my left hand, and here's one strand over here in my right hand. And this finger, these two fingers up here, this guy on the left is a glutamic acid that has a side chain of a COO minus. And this guy over here in my right hand has a lysine with a charge of +1 from an NH3. They're going to be attracted to each other. That attractive force is going to help hold and stabilize this protein, right? If I change the pH drastically and I add protons to that solution such that the COO minus becomes a COOH, it now has a charge of 0. The NH3 is still a charge of +1, but a 0 is not going to be attracted to a +1. I have destabilized the force holding that. It may come apart. Does that make sense? So, very good question. So, what I have just described to you is another force that is stabilizing the tertiary structure of proteins, ionic interactions. Ionic interactions are forces that help to stabilize a protein. Okay, so we've seen disulfide bonds, we've seen hydrogen bonds, we've seen ionic forces. There's another one that's very common. It's known as metallic. And I don't talk much about metallic, but suffice it to say that metallic, some proteins are stabilized by a metal getting in the middle of them and helping to hold them together. Okay? Yeah. Oh, okay, very good question. You're saying break it down to individual amino acids, right? Okay, so his question is if I were to hydrolyze a polypeptide chain, a protein, from its constituent amino acids. So let's say I've got a polymer that's 110 amino acids that are there, and I break it up so it's 110 individual amino acids, and I go back to the original pH, will it come back together? The answer is no. Because that original structure came about because of the genetic code, and they were assembled sequentially in the ribosome. So there's no assembly mechanism, number one, and there's no ordering mechanism, number two, that's there. We'll talk about that next term. Okay? Good. Other questions? It's good. Okay, so the last force that I want to talk about that helps to stabilize the tertiary structure of proteins is one that's a little hard to describe, but you sort of know it. Okay? It's what we call hydrophobic interactions. Hydrophobic interactions. All right? Now, hydrophobic interactions we've already sort of talked about last lecture. And the last lecture, what I talked about was the fact that those nonpolar side chains of amino acids tended to be where on a protein that was dissolved in water. They tend to be on the inside because they're avoiding water. Well, it turns out that when they're interacting with each other, that's a force. That avoidance of water is a force. Okay? Maybe somebody set you up for a blind date, and you really didn't want to go on the blind date, and you heard something about this blind date, and you locked yourself in the bathroom because you don't want to go out with that blind date. That's a force. All right? The hydrophobic amino acids are locking themselves in the bathroom by folding and being in the middle. All right? That is a force. It turns out that that hydrophobic force is actually a reasonably strong force. It helps to hold a protein together. If I want to break the bonds of a hydrophobic force, I have to basically insert something into there that is going to disrupt those hydrophobic interactions. And do you know what we use for that? Soap. The reason we wash our hands is that we are interrupting those hydrophobic interactions, because the long nonpolar tail of the soap interferes with those, it destabilizes the hydrophobic interactions in proteins and allows them to unfold. When they unfold, they are denatured. So again, the reason we wash our hands before we eat and get rid of the germs and so forth, or kill germs and so forth that are on our hands, is because we're denaturing the proteins in them. Just for the same reason that we cook our food, we're killing the proteins in a different way. We're killing them with heat. Okay? So heat versus soap or detergent. So now you know something about the forces that help to stabilize tertiary structure. Nonetheless, you've heard many forces here. Most proteins you got to be pretty careful with. If you've ever tried to isolate proteins in a laboratory, some proteins are exquisitely sensitive to temperature. Some of them are exquisitely sensitive to the buffer in which they're located. And if you've been trying to isolate a protein and keep it pure and keep it active, it's really, really hard to do in some cases. Others, like ribonuclease, you can boil them. One of the ways you can purify ribonuclease is you can boil cells and take the stuff that's left over that still works. That's kind of good. Okay? All right. So, protein structure. Let's see. That finishes pretty much what I want to say about tertiary structure. I'll talk about helix turn helix later in another lecture. I'm not going to talk about that here. And I just want to turn our attention briefly to quaternary structure. So I'll remind you last time that I said that as we get higher and higher levels of structure, we start talking about interactions that are farther and farther away. Primary sequence had interactions between adjacent amino acids because they were peptide bonds. Secondary involved regular repeating structures like an alpha helix or a beta strand that arose from interactions between amino acids less than 10 amino acids away. Tertiary structure interactions are happening between amino acids that are farther than 10 amino acids away, because if I have a very large unfolded protein like I have with myoglobin, I could imagine that this might be amino acid number 6 right here, and this might be amino acid number 60 over here, and that's more than 10 amino acids apart, yet they're close enough to interact, and that interaction helps to stabilize them. So tertiary structure interactions are happening between amino acids that are not close in primary sequence. Well, then you might wonder, how do quaternary interactions arise? They must be not close in primary sequence, and it turns out that quaternary interactions happen between individual subunits. That is, they're separate proteins. Most proteins that we see in cells are actually multi-subunit components. Hemoglobin is probably the best example we'll talk about in the term, and hemoglobin is comprised of 4 subunits. That is 4 separate polypeptide chains. All right? 2 of the chains are identical, and they're called alpha. The other 2 chains are identical, and they're called beta. You see 2 alphas, and you see 2 betas. Alphas and betas are very, very similar in structure, as we will see, and very, very similar in function. Essentially, we will treat them as identical in function. But nonetheless, there are 4 separate units. Well, we start thinking about, well, what's going to stabilize quaternary structure? And it turns out that all of the things we saw that stabilize tertiary structure also stabilize quaternary structure. Hydrogen bonds, hydrophobic interactions, metals, ions, okay, disulfide bonds. All of those can help to stabilize quaternary structure. And so just as adding a detergent to a protein might denature it, so too might that detergent separate and take apart the individual polypeptides of a protein that has quaternary structure. Now, notice all proteins don't have quaternary structure. Not all proteins are multi-subunit. Myoglobin that I showed you earlier is not a protein that has quaternary structure because it only has 1 subunit. It takes at least 2 to interact, right? Hemoglobin, which is related to myoglobin, has 4 subunits. And we'll talk more about both of those later. Okay. So, That's quaternary structure. Here's a nice depiction of quaternary structure. It's actually the viral coat for a rhinovirus, the thing that has caused many of you to have a cold in the past week. Okay? You can see individual subunits. You can see there's many greens, there's many blues, there's many reds. All the greens are the same, all the reds are the same, all the blues are the same. This is an example of something that has assembled itself very, very nicely to encase within it a nucleic acid. Okay? So we have quaternary interactions that are between these individual subunits helping to stabilize this structure. Yes, sir?
What's considered a subunit?
Okay. So what is considered a subunit? A subunit is a polypeptide chain. So I use that term polypeptide chain as identical to protein. So we could say that something that has quaternary structure really has 4 proteins within it. Okay, so the nomenclature is a little confusing. We talk about it being a protein, and then it has 4 proteins, which is why sometimes people say polypeptide units as the subunits. Make sense? Okay. Okay. Well, let's go back and talk about ribonuclease. Ribonuclease is of interest in several levels, and it's also because it gives us some information about the structure of proteins. Here it is again, and you can see the disulfide bonds that I described to you earlier. Ribonuclease is of historical interest because it was the first protein that we determined the sequence of. That is, the sequence of amino acids, the first protein we discovered the sequence of amino acids of was ribonuclease. Okay? I talked about reducing disulfide bonds, and I described it to you, but I didn't show it to you. So let me show you at the chemical level what's happening when we reduce a disulfide bond. Here's a protein that has a disulfide bond, and here is, this is mercaptoethanol. Okay? Mercaptoethanol, you see, starts out with an SH, and you see our protein starts out with a disulfide bond. Basically what happens in this reaction is they swap places. This guy, which has an SH, becomes a disulfide bond, and this guy, which was a disulfide bond, becomes 2 SHs. That's possible because this guy is a reducing agent, meaning that it's donating electrons and protons to make possible the formation of the disulfide bonds. Mercaptoethanol is one of several compounds that we use in the laboratory to reduce disulfide bonds in proteins. Okay? Those another reagent that we commonly use is known as DTT, not DDT, but DTT. It stands for dithiothiotol, and it has a very similar chemistry to mercaptoethanol in terms of what it's doing with donating electrons. The end result is the same, though. When we treat a protein that has disulfide bonds with DTT, we end up with sulfhydryls instead of disulfide bonds. Okay. Now, I showed you earlier that there was urea, and I said that urea had the same effect as boiling because what urea does is it breaks down hydrogen bonds. It interferes with hydrogen bonds. So if I add urea to a protein, there's a very good chance I will denature it because it's going to disrupt its hydrogen bonds, and those hydrogen bonds are going to be necessary for secondary structure, they're going to be necessary for tertiary structure, and if there's quaternary structure, they may be necessary for quaternary structure. So all 3 of those may be critical and may be affected by the addition of urea. Okay? Guanidinium chloride is another reagent that interferes with hydrogen bonds. Also interferes with hydrogen bonds and also will act to help denature proteins. Okay. Now, ribonuclease is a pretty interesting protein, as I said earlier, because ribonuclease teaches us and reminds us about the importance of primary structure, primary sequence of amino acids. So this is related to a couple of the questions that I had earlier, so let me step you through this. Let's say I've got a protein. I've got a ribonuclease. And I do like I said before. I take that ribonuclease and I treat it with beta-mercaptoethanol, and I either boil it or I treat it with urea. Either one works. What I hope you remember from what I showed you earlier is that ribonuclease will not be active. It will unfold, and it'll be in the denatured form. Okay? It's interesting that if I take away the urea very slowly, and I take away the beta-mercaptoethanol, what will happen is over time, I will see a little bit of the ribonuclease activity begin to return. All right? Now, what does that mean? That tells us, as convincingly as anything, that the sequence of amino acids can determine the folding of a protein. Because in order for that protein to become active again, it has to refold, and I haven't put anything else in there to help it to refold. It's refolding on its own using its sequence as a guide. I'm going to repeat that now. So I take this unfolded ribonuclease, I remove the urea, I remove the beta-mercaptoethanol, and this guy will start to refold, and I can see it refolding by the fact that I start to get ribonuclease activity back. Okay? Yes? So his question is that based on the shape of the sequence? And the answer is the sequence determines the shape. So in essence, the answer is yes. Okay? But it's the sequence that's doing it. Okay? But the sequence, because it has shapes in it, are determining that. Okay? Okay. Now, that's really interesting. That tells us, like I said, that the sequence drives that. That's not true for most proteins. You say, well, is the rule not good? No. It turns out that folding is an extraordinarily complex process. Once you've taken things apart and unfolded them for most proteins, they don't have the inherent ability to come back together because their folding was a function of the sequential addition of amino acids as they were being made. That is, they started folding as they were being made, and if you completely take it apart and you put them back together, there's going to be some misfoldings that might happen that would not allow it to come together in the same way as it would if it were being folded sequentially while it was being made. Okay? So ribonuclease doesn't have that limitation. Ribonuclease doesn't need to sequentially start to fold as it's being made. It can come together just from the sequence of amino acids. Most proteins, as I said, won't do that. But nonetheless, this tells us that that sequence there is important. All right? Now, I'll tell you something that will confuse the heck out of you. Okay? I said when I did what I just described to you, which is that if I take away the urea and I take away the beta-mercaptoethanol, I get some of the activity back. I don't get all the activity back. All right? That's a little confusing. And then I'm going to add to your confusion, but maybe, for some of you, give you an understanding about what's going on. All right? If instead of taking away all the mercaptoethanol, I take away most of the mercaptoethanol, but not all of it, I see that I can get close to 100% activity back. Now, there's a puzzle for you. There's a puzzle for you. How come it is when I take away all the mercaptoethanol that I see some of the activity back, but when I take away most but not all, I see almost 100% of the activity return? What's going on? Any thoughts? Yes? She gets an A for the day. Okay? So her answer, if you didn't hear it, I'll repeat it. Her answer is that the formation, if we have a random mixture and a random unfolded protein, the way in which it comes back together, because of that sequential thing I talked about, doesn't necessarily lead to everything forming in the right order. Sometimes that will happen, and some of those you get together. But what if you get the situation like you see on the screen, where you have a red with a green instead of a red with a red? Once a red forms with a green, this guy is never going to be active. But if I have a little bit of mercaptoethanol in there, guess what happens? It allows that to come apart and gives it another chance to fold properly. And that is the answer to the question. Right? That's what happens, basically. That's very cool. Very, very cool. Yes? Good question. Is this energetically favored to be in the native form? Okay? In general, yes, but when we look at the energies associated with folding, there's energies all over the wazoo for each step. And so we have peaks, valleys, peaks, valleys, peaks, valleys, peaks, valleys. It's happening over the folding. And so we can imagine we might, if we started to misfold something, we're not going to have enough energy to get over the next peak in the process. And so, yes, this is energetically favored, but to get to this point is a long path that might have misdirections along it. And that's why most proteins, in fact, don't have this property. They get in those misfolded directions, and they never get to this point. Make sense? Other questions? If you understand that, that's really cool, okay? Because that really tells you an awful lot about protein structure. No questions? You're a quiet group today. Everybody's ready for Friday, and it's only Wednesday. Say it again.
Why did the protein stop in the native form? Like, why do we get all the activity back? Why does it?
Okay, his question is, why does it, once we get it in the native form, why doesn't it unfold again, basically, right? Okay, the answer to that is actually these guys right here. Just like these stabilizers, these guys are stabilizers. Okay? For many proteins, they don't have enough of these in the critical places that they keep it from unfolding. Okay? This guy has enough and in the right places that it pretty much stops that from happening. This enzyme is a real bear if you work in a laboratory on RNA. I see people smiling about that. Do you work with RNA? Anybody work with RNA in here? Yeah. Okay, you know what I'm talking about. RNases are murder. Because if you're working with RNA, you got something that's on your fingertips, you've got something on everything that is chewing up your RNA. You've got to be really, really careful in how you work with those. Okay, that's a cool lesson on ribonuclease. Let's, this is another thing I wanted to talk about. So last time I had some people ask me questions about what happens with the tendency for a given amino acid to be in a given structure. So I promised you a table, and this is the table that I have, that tells you the likelihood, the relative likelihood, that a given amino acid will be in any particular structure. Okay? So here's glutamic acid, for example. The higher the value, the more likely it will be in that structure. All right, so there's glutamic acid, 1.59, very likely to be in an alpha helix, much less likely it's going to be in a beta sheet, and reasonably likely it'll be involved in a reverse turn. We can't look at one amino acid, you can see from this table, we can't look at one amino acid in a sequence and say, what's it going to be in? We have to look at a sequence of them and say, oh, well, here's a stretch of sequence that has an glutamic acid, it has a leucine, it has a methionine, it has an alanine, and we see that they're all lining up nicely with an alpha helix. We recognize that that is very likely to be in an alpha helix. On the other hand, if we saw something that had a leucine followed by a leucine followed by a methionine, followed by a valine, followed by an isoleucine, we would say there's a good likelihood it's going to be in a beta strand. And as I said, we can have a computer program go through and analyze a given protein sequence and say, hey, what's the likelihood that this stretch is in an alpha helix? What's the likelihood that this stretch is in a beta strand? And a computer program can tell you that answer pretty accurately. We have pretty good ideas of the secondary structures of proteins from computer analysis. Tables like this are really invaluable for that. They tell us where the turns are. They tell us where these sequences are. Where these structures are. And we can predict with reasonable accuracy the location of those secondary structures within a given protein structure. What we can't do, and this is amazing, what we can't do is we cannot predict what the overall tertiary structure of a protein is from its amino acid sequence. And people would love to do this. They would love to do this. Okay? It's not possible, for the most part, there are some minor exceptions to that, but it's not possible for the most part to do this because there are so many possible variations in sequence. Not in sequence, but in structure. So let's think about yesterday I talked about the turns. And you have turns that can happen, and you say, okay, well, here's a turn. Well, does that turn have a 60-degree angle? Does it have a 51-degree angle? Is it in 3 dimensions so that it's rotated slightly this way? There's all kinds of possibilities that even slight structures in one turn have big effects down the line as you try to predict where that structure is. So if you miss the first one, then everything else is going to be off. Maybe you get the first one, but you miss the second one, then everything else is going to be off, right? When people first started working with the sequence of proteins, they said, oh, well, this is a simple problem. We know the chemical structures, we know the dimensions, we know some of the angles and so forth, so we can predict the overall structure of a protein if we get just a powerful enough computer. And there were a lot of people that spent a lot of time trying to do just that. And there's people today still working on it in a different way. But what they quickly came to realize was that no matter how hard they tried, they weren't getting anywhere. They weren't able to make very accurate predictions of tertiary structure. Secondary structure, yes. Tertiary structure, no. Their predictions were way off. And they know they're way off because we can actually determine what the structure is if we do what's called X-ray crystallography, where we shoot X-rays through a protein and we can tell where those atoms are. But it would be nice if we could predict it where we didn't have to shoot X-rays through it and do all that, so people work on it very hard. What I'm getting ready to describe to you is what's called the protein folding problem. It's also called Levinthal's paradox. Okay? Levinthal's paradox. This is going to blow your mind. It blows my mind. All right? Let's imagine we've got a simple protein. It's 50 amino acids long. That's relatively short as proteins go. 50 amino acids long. It's got 50 peptide bonds in it, 49 peptide bonds in it. All right? We start thinking about all the different possible angles and all the different possible interactions that each of those guys could have. And Levinthal did this and came to the conclusion, okay, that if he looked at all the possible angles and started that if protein folding was occurring by a random process where it went through every possible configuration before finally ending up with the right one, if it went through that process, Levinthal looked at it on a computer and says, oh my God, right? The possibilities blow my mind. It can't be a random process. I'll tell you why. What he discovered was, considering all those possible different structures, that if it were on a random basis and they were to go iteratively through and analyze every one, if you took the most powerful computer on Earth that's available today and you had it analyzing a structure every nanosecond or so, okay? So you have all these different possible structures that you can analyze on a very, very rapidly working computer. This is a very simple protein. How long would it take to go through all those possible structures and determine the ultimate correct structure? Well, it turned out that computer couldn't do it. And they said, well, what if you took all the computing power on the face of the Earth and you applied it to the problem? Could you do it? And the answer was you couldn't do it. What he discovered was if you took every computer on the face of the Earth, and they were all the most powerful computer on the face of the Earth, and they worked in this problem forever, it would take 1,000,000 times longer than the age of the universe to do it. It's probably not going to be solved by a random basis. Okay? That tells us a couple of things. One, we don't want to use random methods for trying to identify structure, and we know that. Number two, it tells us that protein folding does not occur by a random basis either. We may not understand all of the things that are happening in protein folding, and yes, we're making good progress and learning a lot about how those things are in fact happening, but they're not occurring by a random process because a protein inside of your cells can properly fold in a matter of seconds. In a matter of seconds. And fold the same way every time. Or almost every time. Okay. Pretty cool stuff. Now, we also know that folding is a very, very critical process in our cells. What you've seen is that it doesn't take much energy, and in some cases it doesn't really take any significant energy for something to not fold properly. You saw that with ribonuclease, right? Even ribonuclease didn't come back together 100% of the time unless I added mercaptoethanol. I had to give it more chances to come back together properly. Some of those chances didn't work out right. In the case of ribonuclease, no consequence. No consequence because it means that ribonuclease simply isn't active. There are proteins in your brain for which there's enormous consequence if they don't fold properly. Enormous. Everybody in here has heard of mad cow disease. Okay? Mad cow disease is not caused by a bacterium. It's not caused by a virus. It's caused by a protein that's found in every one of your brains. My brain, the cow brain, they're all there. Every one of us has this protein. And when this protein that we have in our brains doesn't fold properly, we're in deep doo-doo. We see it in mad cow. We also see it in human diseases known as Creutzfeldt-Jakob. And if you ask me later, I'll spell it for you, but I won't spell it here. Okay? Creutzfeldt-Jakob syndrome is almost identical to mad cow. It's a neurological problem, and people who develop this disease last a few months before their brain is gone. Dementia out the wazoo. Okay? It's happening because this protein that was in their brain, one of them misfolded. Didn't fold properly. Okay? It didn't fold properly. And as a result of it not folding properly, this is the bad part of it. You say, well, the ribonuclease didn't fold properly. No biggie, right? Well, this protein, it's a biggie. Because this protein, once one of them doesn't fold properly, it causes others of the same nature to also not fold properly. This protein is called PRP. Here's the normal PRP. It's a normal brain protein. You have it in your brain, and here's its proper folding. Here's one that started out not folding properly. What one misfolded one can do is cause others to misfold, and they form large amyloid plaques that destroy nerve cells in our brain. They destroy them very rapidly. That's why Creutzfeldt-Jakob works very rapidly and causes dementia very quickly. Okay? It's not completely known why it misfolds. It's not completely known, for example, if it can be transmitted or how easily it can be transmitted. It does not appear to be easily transmitted in humans, but there's some evidence that it can be. People who work, for example, with brains of dead people who suffer from this disease sometimes have an increased incidence of developing it. There was an incidence of mad cow disease, an outbreak of mad cow disease in the late 1980s in Great Britain that was well known, well documented, and at that time they didn't inspect British beef, and so it made it into the food supply. And about 10 years later, they saw a pretty good bump in the occurrence of an odd form of Creutzfeldt-Jakob in humans. The concern being, can it be transmitted in the food? It's a controversial issue. Some people say yes, some people say no. But here's the really bad news. Cooking it doesn't protect you. Okay? This protein is extraordinarily stable. If you want to completely denature it so that it can't cause effects, you've got to heat it up to about 700 degrees Fahrenheit. I don't know any recipes that call for cooking at that temperature. Okay? So, that's a concern. A real concern. Okay. Questions, comments about that? Yeah, once someone has it, can you do anything about it? Can you unfold the unfolding is basically what you're asking. I know of no single incident or no single occurrence where a person has been cured of Creutzfeldt-Jakob. No. It's a disease that is prevalent throughout the animal kingdom. Probably every animal has it. I mean, that is, there are animals who have it. For example, I mean, beef is one, but there's a disease in sheep called scrapie that is caused by the same thing. It's known to be in deer. It's known to be in a variety of things. It probably is in virtually every animal that's out there. And there's evidence that it may, in fact, be a problem throughout the kingdom of life. There are misfolded proteins that cause some unusual problems inside of yeast. Okay? So, yeah, that's kind of a scary thing. It is. Okay? Now, the last thing I'll tell you, and then I've got a fun thing at the end. The last thing I will tell you is that folding of proteins is really critical. All right? I hope I've convinced you with this, but our cells know they're very critical, and our cells actually have things in them to help proteins to fold properly. They're called chaperones, or chaperonins. You can call them either one. Chaperones are structures that proteins get put into to allow them to fold properly. You might think, what in the heck does that mean? Well, they're basically a little chamber. All right? Let's think about a protein being made. A protein is being made one amino acid at a time. And if I think about a typical protein that's dissolved in water, I think, well, there are hydrophobic amino acids there, right? And if I have hydrophobic amino acids there and they want to fold away from everything else, what happens before they fold? Do we have problems? We might. They don't like water. They want to get away from water, and they'll interact with anything that allows them to get away from water. So imagine I've got this protein being made over here, and I've got a few hydrophobic amino acids that come off, and I've got the same protein, another copy of it being made over here, and it's a little bit further along. They're close to each other. The hydrophobic amino acids of this protein might interact with the hydrophobic amino acids of this protein, and thus prevent proper folding. What chaperones do is they allow the proteins to be isolated and fold on their own so that they don't have that interaction with each other and misfold. That turns out to be really, really useful. Okay, I promised you something fun, and let's finish with something fun. It relates to the fact that I'm tired of all this sunshine. I'd like you to think about that. Oop. Ah.
On the Oregon weather's cloudy, the sky is mostly cloudy. You can't stop it, you complain. So let it rain, let it rain, let it rain. It doesn't show signs of snowing. It's rarely right for snowing, though it's driving some folks insane. Let it rain, let it rain, let it rain. When it finally turns out dry, we'll be putting away our reindeer. It'll probably be July, but I'll surely miss the reindeer. 'Cause the sound of the pouring rain, when they're pattering down the drain, it's music inside my brain. So let it rain.
All right. See you guys on Friday.